This is a demonstration of the solid sample cell used for determining surface zeta potential of for the NanoPlus and NanoPlus AT. This product is sold by Particulate Systems, which is a division of Micromeritics Instrument Corporation. And as I'm taking off this top layer, this type of cell is used to determine surface zeta potential, as I mentioned, of things like membranes, plates, coated materials, any kind of flat surface that comes in contact with a liquid. So we'll take this top part off, and I'm going to demonstrate how to disassemble and then assemble this cell. So I just took off this top part. This middle part I'm going to take off so that I can place the glass cell inside. The glass cell has a coated surface, so it's actually stored in water. The coated surface is used for keeping it moist. It has a neutralized surface so that it doesn't interact with the monitoring particles. This way, when we're making the analysis, we're only looking at interaction with the upper surface, which is the sample. So we'll remove that, and then here on the inside you can see the electrodes, and you don't necessarily have to do this, but for completeness I'm going to go ahead and take off this bottom layer so you can see the electrodes in case you have to clean them. This cell uses electrophoretic light scattering to determine the surface zeta potential. So you see the O-rings here, in case you need to replace them. There's spares that come with the cell. The electrodes are here, and we can pop them out. They look like little coins. There's a dull side and a shiny side. When you place them back into the cell, the shiny side goes down, and there's a little groove etched in here, so the electrode should fit perfectly in the same position each time. So that's good. We'll replace this piece. You do get also spare screws in case you lose one of those. Typically I'll just kind of screw them on just a little bit and kind of run my finger across, and then use the torque wrench to kind of tighten them. You can't over tighten because, like I said, it's a torque wrench. You just want to make sure that they're secure. That way we don't have any leaks when we assemble everything. This is the glass stored in water. You see it's also wrapped in a tissue, lab tissue, so that the water stays surrounding the entire surface to get it out. And then we'll rinse it with just regular water is fine. And you want to take care of these edges. The long edges, that's where the light path will be. And also on the top part there's a little groove, a rectangular groove. The sample has to fit over that entire inner rectangle, and this rectangle actually has to be facing up top. So once we have everything cleaned up and ready to go, you place it into the cell like this. Make sure that it goes down flat, just like that. And so you have this opening, this groove facing up, and you have the light path clear of debris. and then we'll place this middle piece back on to hold the glass in place. One of the unique things about this cell also, besides monitoring the surface zeta potential, is that it has this inlet and outlet where you apply the monitoring particles, and this allows it to also connect to the autotitrator as if it was a flow cell. And in doing so, you can monitor surface zeta potential as it relates to pH or additive studies, changing salt concentrations, things like that. This is really important for the membrane people that use membranes for filtration. You could determine the surface isoelectric point, or the pH where the surface zeta potential of the membrane is 0. So now we have this, and then we're gonna put our reference material. This is PMMA, polymethyl methacrylate. Take it out of the box. It's just a piece of plastic. You're gonna peel this off to expose the analysis surface. So you have the surface that's for analysis here, then you have a numbered surface on the top. We're gonna place that down over that groove so it fits just over it. And then we'll place our spacers in place. Since this is a hard surface, we're going to use one of our soft spacers first. And then we'll use a more rigid one. And just for extra, like I said, these are just spacers that don't do anything else than that. And then occupy space, we'll just put another one there. And then this cover, that when it goes over, this lock is a little kind of like elevator. It'll just go down and clamp down onto the sample surface. So I'm going to screw this on. Actually, I don't want to tighten it just yet. I want to wait till it's a good habit to get into just finger tightening everything first, and then go back with a torque wrench and tighten them all at the same time. So that way you don't have any bulges. If you over tighten one side versus the other. So we turn this lock, and again it comes down and presses firmly against the spacers, which press down on the sample, holding the sample in place over that analysis chamber, that groove that's inside the glass piece, so that you see where the path length of the laser goes through here and here. Now we have to make our monitoring solution. So I have deionized water here. For better results, you'd want to use 10 millimolar sodium chloride, but if you use water, you should be okay. And the monitoring particles is just some latex spheres. It's a very concentrated suspension, so you'd want to make a dilution of 200 to 300 times. I typically take about 2 to 3 drops in about 20 ml of water. And the goal is you just want to get that water just slightly turbid. Just shake it. So that's ready. We'll use syringes to fill it. If we were connecting this to the autotitrator, this is the solution that we would be titrating and flowing into the cell. And then once we have everything together, we'll press down and you'll see the other syringe come up. That means that there's no leaks anywhere. So no matter how hard I push down, it's going to push up in here, so the entire system is sealed. And I'll look inside to make sure that there's no air bubbles moving along the surface of the sample. Air bubbles will interfere with the measurement. And once we're done with that and we're satisfied that there's no air bubbles, we'll pull the syringe off and put these little lure plugs in place. And that's it. It's ready to go into the instrument and start the analysis. The instrument will do a cell center and then do a 7-point measurement, profiling the top to bottom of the cell, and from that we'll determine the mobility of the particles at the different the layers, and determine the surface zeta potential.